So hello and welcome to another video in the lecture series entitled Advanced Biomaterials. So in this series we are looking at the different scaffolds or what are scaffolds and their uses in tissue engineering. So in the previous video we had seen about the scaffolds which are used in tissue engineering, their properties and their characteristics and some of their important features which enables them to be used as essential components in tissue engineering and other advanced biomedical applications. So in today's video we will be looking at some of the important scaffold fabrication techniques. So let's get into it. So different materials have been used as for making scaffolds for tissue engineering. So some of them are the different types of biomaterials. They are classified as natural and the synthetic ones. So on classifying the materials for scaffolds, so these are mainly the natural and synthetic ones. And synthetic. Now the natural sources or the natural polymers which are used for making these scaffolds are include the components of or the whole extracellular matrix which is obtained from allogenic or xerogenic sources. So on a whole, the extracellular matrix can be used to fabricate natural types of scaffolds. So extra natural scaffolds can be obtained, modified and tailored from natural sources. So they can be processed such as the so from allogenic or xenogenic extracellular matrix. So allogenic and xenogenic extracellular matrix which can be used as a suitable natural scaffold for tissue engineering applications. So allogenic, as the term suggests, it is from genetically dissimilar individuals, but individuals from the same species. So and xenogenic means individuals from the different species. So from these, obtaining the extracellular matrix can be used for fabricating scaffolds for tissue engineering applications. Now the main different proteins and organic polymers can also be used in such cases, which include the proteins and the polysaccharides. So the main disadvantage of this method is the limited mechanical and physical stability of the scaffold. So limited mechanical and physical of these scaffold systems, and also it has another major disadvantage or major drawback, that is such materials can be immunogenic in nature. So that is why it might be so since these materials might be immunogenic in nature, so these might be immunogenic to the host, because these are external proteins and this might act as antigens when used inside another host tissue, because these are from different individuals or even from different species. So that is why these instances need to be considered while choosing such materials for fabricating natural scaffolds. So how can these be corrected? So for the first one, for improving the mechanical properties, so it might be possible to reinforce the materials to the desired mechanical properties. So the necessary reinforcement of the mechanical and shape stability of the natural biomaterials can be performed. So this would be these examples would include developing composites with synthetic material or varying the cross-linking. So developing composites, so composites can be used to correct these to improve the mechanical properties through reinforcing materials for improving these characteristics, and also for by cross-linking, improving the cross-linkings as we have discussed previously. So by changing the cross-linkings, it might be possible to correct these defects. And in order to alter the immunogenicity, there might be suitable types of coatings or treatment with collagen from the host tissue, or other types of suitable protein-based treatments to improve the immune response in the host materials, in the host body. And now coming to the synthetic category of these scaffold-based materials, these can be again divided into two types. The first one is inorganic, examples of which can include the bioglass, and then the organic, which can include the different polymers. So then the main disadvantage of the synthetic type is the biocompatibility. So coating, this can be improved by coating the natural biomaterials with materials such as collagen to improve the biocompatibility. Then also, but with the synthetic biomaterials, based materials for fabricating such scaffolds, it is possible to better control the physical and mechanical properties, can be tailored for both hard and also for soft tissues. So better tailorability for hard and soft tissues is possible through the synthetic scaffold materials. So that is why synthetic materials are more preferred over the natural types of materials for fabricating these different scaffold constructs. So now let's come into some of the scaffold fabrication techniques. So the scaffold fabrication techniques can be mainly classified into three headings, as if we discuss it. So the first strategy is known as the process which uses porogens in biomaterials. So process which involves or uses porogens in the biomaterials. So what are porogens? Porogens are materials, or porogens are actually agents which causes the formation of pores in a given material. So scaffold materials, as in solids or dissolved in solvents, are incorporated with porogens like gases such as carbon dioxide, liquids such as water, and even solids such as paraffin. Now these mixtures can be then cast, processed, and extruded. So after this, after fabrication, these porogens can be removed using appropriate steps such as sublimation, evaporation, and melting to get the porous scaffold. So then after incorporating the porogens, then these porogens are actually removed through sublimation, evaporation, or melting processes to leave behind a porous scaffold. So after removal of the porogen, the porous scaffold is obtained. So the examples of these techniques would include, let me remove this part so we can write the examples of this technique. So these include the processes of particulate leaching, Then there is the example of gas foaming. Then one of the mainly used or one of the easiest scaffold fabrication techniques, which is freeze drying. And then we have the phase separation techniques. So particulate leaching, gas foaming, freeze drying and phase separation techniques are the common processes which uses porogens for creating scaffolds in this approach. So now let's come to the second fabrication technique, which is essentially number 2, which a technique which uses solid free form or also known as the rapid prototyping technique. So the second technique is the solid free form or the rapid prototyping technique, wherein hierarchical porous structures can be manufactured by sequential delivery of material and or energy needed to bond the material to present points, bind the materials to present points, preset points in space. So there will be a hierarchical porous structure which can be manufactured through sequential or through a controlled delivery of the material or energy needed to bond the material, to bind the material to preset given points in space. So this method can be better understood through the examples which include the selective laser sintering. So one includes the selective laser sintering, then the stereolithographic methods, which is being recently used for fabricating different types of scaffold based materials. So stereolithography, and finally again one of the most convenient or one of the most widely applied processes for fabricating different types of scaffolds with very controlled pore sizes, in which can be preset from different points, or which can be actually predetermined from different softwares through the 3D printer, which is known as the 3D printing technology. So 3D printing techniques. So these methods can be used in the classification of solid free form or rapid prototyping techniques. So in this 3D printing technology, we will be discussing this technique in some of these upcoming videos. So this is a very interesting technique which can be used for fabricating different types of scaffolds with controlled or predetermined parameters such as port size, port connectivities, for making scaffolds for a very specific type of application. So finally, the last fabrication technique is which uses woven or non-woven fibers, is the third one. So woven or non-woven fibers. So herein, woven or non-woven fibers are actually piled together and bonded using thermal energy or adhesives to give a porous meshwork using techniques such as fiber binding. So these fibers can be generated through the common process which is known as electrospinning. So with electrospinning techniques, so these fibers are actually used for fabricating. Through these fibers are actually given, which are then wrapped, or which are then connected or bound together or spun together to give this a porous network or this porous meshwork of this scaffold. So electrospinning comes under this type of scaffold fabrication. So based on the particular applications of the scaffold which we are trying to fabricate, or based on the location or the tissue type in which that fabricated scaffold is used, is intended for its use, so we can select both the material and the method for fabrication of the scaffold based on this type of a specific requirement. So some of these, including the one of the most important, so freeze drying method in the Porogen-based based scaffold synthesis technique, and 3D printing in the rapid prototyping technique, and in the fiber utilizing technique, electrospinning. These three are some of the most commonly used scaffold fabrication techniques which is used in different labs for fabricating scaffolds for different applications in tissue engineering, such as skin regeneration, bone tissue regeneration, templates, chondrocyte injection and cartilage repair, and so many nerve tissue repair, so many wide range of applications where these scaffolds play a main role in tissue engineering. So in the upcoming videos we will be looking at some of the important examples of actually how scaffolds are fabricated for treating a particular condition. What are the different parameters which needs to be considered in a specific case by case study for designing a particular therapeutic intervention for restoring or supplementing a tissue type or function. So with this we will end today's video. Hope you enjoyed it. See you in the next one. Till then, bye.